students, here move to chapter 2, activity 2 about mitosis and meiosis. First of all, let's have a funny idea, let's say, about biology. Biology is the only science where multiplication and division, they have the same meaning, or they do the same thing. For example, mitosis. Let's start with mitosis. Mitosis is the process by which somatic cells, diploid cells, two end cells of the body, they produce new daughter cells. They are identical to the mother cells. Why would the cells need to reproduce? First, to increase the cellular number. Second, to renew body cells, tissues, and replace the old cells. Mitosis is composed of different checkpoints and different events. It may be divided into interphase, G1 as G2, gap 1 phase, S phase, transitive phase where DNA duplication or replication occurs, and G2, gap 2 prepare itself for mitosis. Mitosis is composed of four phases: prophase, metaphase, anaphase, and telophase. So this is the cell at the interphase, where we have the chromatin form. Here we have the centrosome that will change into asters, the nuclear membrane, the nucleolus. So in the mitosis, first we have interphase, the preparation. Second, we have prophase. Prophase is the cell where chromosomes, they are composed of two chromatids. Centrosome, they are going to change into aster. Spindle fibers start to form. Nuclear membrane disintegrates, and the chromatin condense, as I said, into chromosomes, each made of two chromatids. Second, we have metaphase. In the metaphase, the chromosomes, they are in the middle of the cell. They form what's known by equatorial plate. Anaphase, each chromosome is going to be separated to opposite poles of the cell, where sister chromatids move to opposite pole of the cell as seen in this figure. This is called a polar ascension. So sister chromatids, you are pulled by spindle fibers. Each chromatid moves to the opposite pole of the cell, forming what's known by the polar ascension. Late anaphase, the result will be two cells, each made of one chromatid. Chromosome is made up of one chromatid at the opposite pole of the cell. Last phase is telophase and cytokinesis, where we have the cleavage furrow. This is called the cleavage furrow. The chromosomes form identical groups. These chromosomes will decondense back into the chromatin. The nuclear membrane starts to form around each set of chromosome. The spindle fiber disintegrate. You have to know that the constriction of cleavage of the cell is referred to as cytokinesis. We have to know that two daughter cells obtained after every cycle are identical to each other and identical to the mother cell. These are different figures at interphase. This is a prophase, metaphase, anaphase, and finally we have the telophase. We have the cleavage furrow, as I said. Cell at the end of interphase is like this. So meiosis. Meiosis is the process by which mother cells, they produce gametes, the primary sexual organs. Before meiosis, the mother cell of gametes will undergo interphase. Then it's going to undergo into two divisions: reduction and division. Meiosis I, the amount of chromosomes and genetic information is reduced into half, and the meiosis II, equational division. So let's start with meiosis I. Meiosis I, prophase I, here we have a crossing over or synapses. Here we have nuclear membrane disintegrate, the same as mitosis. Mitotic spindle fibers start to form, the same as mitosis. Centrosomes change into aster, the same as mitosis. Chromosome they condense so that each chromosome is made up of two distinct chromatids. All of them, they are similar to mitosis, but here we have the difference. Homologous chromosome, they pair up, they synapse, forming what's known by tetrads. This will increase the chance of the crossing over. Here are the tetrads. Second, we have meiosis I, metaphase I. Chromosome, they align at the middle of the cell, forming what's known by equatorial plates. And anaphase I, the chromosomes will be two groups, each made of two chromatids at the opposite pole of the cell. Polar ascension, the key word here. And telophase 1 and cytokinesis, the cell is going to constrict into 2, the spindle fiber disintegrate and the aster return to the centrosome state. Prophase II of meiosis II. Similar to mitosis, myostatic spindle fibers start to form, centrosome change into aster, nuclear membrane disintegrates. Metaphase II, the same also as mitosis, the chromosome they line up in the middle of the cell, forming what's known by equatorial plate. Anaphase II. In Anaphase II, sister chromatids are pulled apart. Same process. So mitosis and meiosis II, they are the same process, but at the end, 2 cells. In mitosis, 4 cells at the end of meiosis II. Telophase II and cytokinesis, the results will be 4 the cells, 4 daughter cells. So difference between somatic mitosis and meiosis. Mitosis, it occurs in somatic cells, while this occurs to the mother cell of gametes. It aims for the growth, renewal, or replacement of old cells. It aims for production of gametes. 1 division only, 2 daughter cells. 2 division, 4 daughter cells. Mother cell is 2N and daughter cells are 2N, while mother cell 2N, gamete they are 1N. Made up of 1 division preceded by interphase, made up of 2 divisions preceded by interphase. No pairing of homologous chromosomes in mitosis, pairing of homologous chromosomes in mitosis. Daughter cells obtain 2N, 1 chromatid per chromosome, 2N first 1 and 2 chromatids per chromosomes after meiosis I, after meiosis II, 1 and 1 chromatid. Meiosis II, as I said, is greatly similar to that of mitosis.